We're recording. Okay. We're not there yet. All right. Oh, that's okay. We're actually planning on making this one a little bit short because I also have class at 1:30. So not the full 2 hours. And obviously it'll be recorded. But thanks for stopping by anyway. Um, Once again, hello, everyone. We just had a really cool class this morning. And we wanted to ask you some questions about the stuff that we learned this week. Pretty light week, not any dense PowerPoint slides, just some neat presentations. Um, so let's talk about it. Firstly, we had Dr. What was his name? Stefan Prost, and he talked about conservation genomics, specifically his work on cheetahs in Africa, and what else was it? The stuff in the rainforest. I don't remember exactly which species it was, but yeah. So conservation genomics and rhino. Thank you, Kinga. And I wanted to ask you a little bit about something that we have to worry about in conservation genomics. And it's with this graph. What has happened to the population that is represented on this graph? You can look at the axes. See that on the y-axis is the inbreeding coefficient. Then on the x-axis is the time. Plateauing. I wouldn't say so. Like that's the shape, I guess. I'd say it's more logarithmic, but inbreeding depression. Yes, Joseph, and what does that signify? What could have happened to this population? Homozygosity increased. Oh, great job, Maya. Thank you. Yes, so this was actually the graph of the population and the graph of heterozygosity among the population of the Florida panther from 1950 to 2010. And, panthers or cougars, excuse me, humans are scared of them for some reason. And so they'd kill them whenever they saw them. And so their population dipped so low from 1950 or starting at 1950. And over time, they had extremely high amounts of inbreeding that we can see. The inbreeding coefficient went from virtually none in 1950. Like there was no inbreeding because they had such a large population. Then over time, it grew to almost 80% inbreeding just before 2000. But then in the year 2000, we had an extreme inbreeding, right? Maybe that's not the right term, but just like Maya said, we had an extreme reduction in the amount of inbreeding that was occurring. And that's because of the introduction of the Texas panther that was allowed to interbreed with the Florida panther. And so because we had such, well, maybe not such a diverse genome, but relatively diverse among such a highly inbred population, that the introduction of this distant enough relative from Texas allowed a large jump in the amount of heterozygosity that we saw throughout the panther genome. And so we can see the almost instant dip from 80% inbreeding to about maybe like 6, 7, 8% inbreeding, even by like 2001 or 2002. So that's what happened. Very cool. And who can think of an example of this happening that we saw in Dr. Post's lecture? The cheetahs. Did that happen with the cheetahs? Honestly, sorry, I wasn't paying attention. But I thought that's what happened with the lynxes. Maybe? Question mark. Anyway, yeah. Still relevant. Still cats. And we can move on now, unless anyone has any questions about this. Excuse me. 2 questions. Nope. So the graph. I have a question. What's up? Yes, I think I misspoke when I verified or validated your answer earlier. So inbreeding depression is, or should we say inbreeding decreased, inbreeding increased, then decreased? Yes. Inbreeding depression refers to the reduction of heterozygosity. This graph is a direct graph of inbreeding itself, but usually when we see graphs, you'd have like heterozygosity, also known as diversity. Uh, oh, does someone have a question? Someone's unmuted. Maybe not. Okay. Oh, no problem. Sorry. Yeah, so usually we'd see graphs like directly graphing the amount of diversity that we have in the genome of a population over time. Uh, and then if we saw that or goodness gracious, heterozygosity take a dip, that would be an inbreeding depression, most likely. And that depression refers to the reduction in heterozygosity or diversity. But this graph here is a direct graph of inbreeding of individuals. So the inbreeding depression that you'd see on a heterozygosity graph is actually this increase from 1950 to 1999 or whatever. That would have correlated to an inbreeding depression within this population. That was a really roundabout way of saying it. Yeah. Excuse me. So more about that, like how can we save a species? We did just allude to one example earlier. So inbreeding increased for 2000, then decreased after. So this graph shows on the y-axis inbreeding coefficient. The higher the inbreeding coefficient, the more individuals within the population are inbreeding, and that leads to, a reduction in the amount of diversity within the genome. So from 1950 to 1999, we see an ever increasing amount of inbreeding, which is probably not the best. Yeah. So how can we save a species? And if you can think of any methods, what are the limits to them? We can do outcrossing. That is one, where we create a new species, pretty much by hybridizing our endangered species with a different species that might also be endangered, but might also be just fine. But what is the drawback to that? That's very true. There's no guarantee that crossing two individuals who are from a different species, and if we can think back to Bio 213, what is what is speciation in the first place? The production of species. Actually, no, this wasn't 213, this was EcoEvo. But the production of species, or being a different species in the first place, via the biological species concept, is the idea that you have two reproductively isolated populations of individuals. And even if they might be, excuse me, like 99.98% genetically identical, if their gametes can't combine to produce a viable offspring, then they're reproductively isolated and they're different species by the biological species concept. Excuse me, goodness gracious. So yeah, that is one drawback. That it might not always work if you try to outcross individuals. Another one is that, one that I could think of is that if you outcross an endangered species with a non-endangered species, you're not like creating more individuals of that endangered species. You're just creating a new species. So like while you are passing on the unique genes of the unique species, you're not really preserving that specific species. Any other ideas? Relocation and new habitats. That's a great idea. Yeah, if we have organisms that, let's say, stinky humans come along and destroy their natural habitat entirely, And we just take them to a new place so that they can have a non-destroyed space to inhabit. But what are the drawbacks to this? I can think of several. They might not adapt. It might be traumatizing, that's true. So they simply might not adapt, perhaps because they're traumatized by being relocated. That's another very good point, Joseph. Is that so? Firstly, they just might not adapt to their new environment. Maybe the climate isn't exactly the same. Maybe the other species that they depend on to survive aren't present in the quantity or even at all that they need, possibly. And Joseph is getting at a really good point that there are going to be new predators in that new environment. But what I was thinking is also that the population we introduce could be an apex predator in that new environment where one previously didn't exist. And so we introduce this species to save them, but then they cause a whole bunch of havoc by becoming an invasive species. What about the food source? Exactly. They could over consume a native food source in their new area that other animals depend on, and that limits the amount that's available for the species that already existed. So relocation is, unless it's like a perfect fit, and like it has to be an absolutely perfect storm, and that almost never happens. I can't think of a relocation event that's ever been successful. But another one that I was thinking of was, what if you have an organism that is in a perfectly unique environment where you can find it in no other place on Earth? That habitat's destroyed, and then there's nowhere to relocate them. There is no identical environment for them to inhabit. So, there are a whole bunch of blocks, exactly like polar bears. Like, where can polar bears go when anthropogenic climate change uh, melts ice at the poles? Like, where do they go? There's no more ice for them to camouflage and hunt, you know? So, very good ideas. Very awesome, guys. Thank you. Um, and then a follow up question: what can we do if there are no living members of the species to save? We talked about like how we can save someone that's on the brink, but what if there's none left? Oh my God, are we recording? Ah, please. Okay, thank you. Oh my God. Ah, okay, Joseph says, extinct we are. Thank you. And thank you, Kinga. Did anyone have any other ideas? I can't really scroll up with the chat. Polar bears, nothing. Maya says nothing. Well, I can think of one thing, but for the most part, Maya, you're right. Like a Jurassic Park type of thing. Exactly. And that's kind of, well, I've never seen Jurassic Park, so honestly, I'm not sure. But what I think happened in Jurassic Park is what happened with the black-footed ferret. Someone has a comment, or conserve a similar species. That's true. Obviously, like sometimes it is just too late. Humans have caused such a mess that they're gone forever, and there's nothing we can do. But in this case, we found one living individual a long time ago, and then they became extinct. But let's see what happened.
Against the backdrop of a Colorado conservation center, the romance of the century is about to unfold. In 2022, researchers will attempt to breed Elizabeth Ann, the world's first cloned black-footed ferret, with a captive
Can you hear
male.
this?
If successful, their offspring will infuse the critically endangered species with a splash of genetic diversity. Cloning for the sake of conservation isn't unique to black-footed ferrets. Another endangered species clone is a Przewalski's horse named Kurt, but Kurt isn't yet ready for breeding. And other earlier clones of Bantang and Gower were essentially proofs of concept. They were never bred, limiting their genetic relevance. The lack of genetic diversity is a big problem in re-establishing a species population. If captive animals are too closely related, they're at risk of inbreeding. That can hobble their reproductive fitness, stymieing
Oh, yeah,
family
sorry.
trees. Cloning can help bypass this problem, bringing into play a more genetically distant relative to breed with animals already in captivity. In Elizabeth Ann's case, scientists took cells grown from a skin sample of a long deceased black-footed ferret, preserved in test tubes at the San Diego Frozen Zoo, to craft an embryo, then transferred it to a domestic ferret that brought the embryo to term. This process, somatic cell nuclear transfer, has been used for decades to duplicate livestock and pets. Cloning for conservation, meanwhile, has been on the back burner for a few reasons. Breeding practices are less developed for species in need of conservation, and there's less commercial interest in maintaining endangered animals. Also, cloning for conservation has a tricky endgame. To keep a species as genetically pure as possible, biologists prefer the clone's family tree contain only the genes of the endangered species. Animals cloned by somatic cell nuclear transfer still carry their domestic mother's mitochondrial DNA. This means the offspring of a cloned animal, such as Elizabeth Ann, stand to bring domestic genes into the endangered population. But because mitochondrial DNA is inherited from an animal's mother, these domestic genes can be ferreted out by breeding the male offspring of clones with captive females. If successful, the process could make an endangered population more resilient and eventually lead to self-sustained growth. Just over a month removed from her first birthday, Elizabeth Ann remains a marvel in her own right. If she finds her captive male suitor to be just as marvelous, their once-in-a-lifetime romance could propel their species for lifetimes to come.
Very cute.
Grammarly does more
Leave
than
me
catch
alone,
errors.
Grammarly. Go
With
right.
Grammarly, you can find really good, no, perfect words that make your writing.
Stop. Okay. So yeah. What can we do in the event, in the event of an endangered species? Well, hopefully we have some DNA of the last living individual, and then we can clone them. And what we have done is that we introduce these extinct genes, or the genes of individuals that are no longer living, present in a population and available to reproduce naturally. We can inject these genes that we're trying to save into a population that is close enough so that we can preserve, like, at the very least, some of them, some of the special genome fragments stuff. You know what I'm talking about. Yeah. And whoever like names these ferrets should get a raise at work. Like Elizabeth Ann is just too cute. But yeah, any questions? I know Grammarly follows me everywhere. Go away. Nah, I don't like installing stuff. Because what if it goes wrong, or what if it slows down my computer? I'll consider it, but I guess no one has any questions. Are these the type of questions that she would ask on the exam based on the guest speakers? I'd think so. Dr. Chiari and I don't share a brain, so I'm not like exactly sure what she'd want or what she'd even ask. But like these reviews Sam and I make are like we talk about the concepts that we think are the most vital for understanding. So I'm not entirely sure that these are what Dr. Chiari would ask, but it's what I would study if I were in your position. And I was. Yeah. Okay, so if that makes sense, then we can move on.
All right. My questions are not nearly as involved as Garrett's at all. They're just kind of like asking about like a couple of quick concepts that we went over during class today. I like really enjoyed today's lecture. I thought it was super interesting. Garrett, I don't think I can share my screen.
Sorry, let me fix it.
But is there, I only have two questions and they're like super quick. So let me know if there's anything else that you would like to go over after we go over these questions. So first question, what do proto-oncogenes do? And I can't remember if you guys talk about this during lab or not, but I do remember talking about it during lecture when I took the class. But anyways, nonetheless, what do proto-oncogenes do? Let's answer that question first, and then we'll get to what happens when they become mutated. Yes, it is related to cancer. So basically, what happens is proto-oncogenes are involved in normal cell growth. So basically, they are, yes, they turn on oncogenes. So basically, they're just making sure that our apoptosis is happening correctly. So like when we learned about mitosis, we talked about how we have all of the different checkpoints and stuff in the cell cycle. And so proto-oncogenes, what they do is they will actually help cause apoptosis during those checkpoints if something is not right with our cell. So I guess that kind of sort of, I gives like a hint to the next question. So what happens when we actually mutate them? Yes, we do get cancer. So basically what happens is we are no longer able to cause apoptosis to for like cells that do have like some kind of mutation or something wrong with them. So like, for example, if a cell has didn't replicate its, or like if it has like 2 times as much DNA as it's normally supposed to after S phase, then obviously like your cell, the cell cycle would stop and be like, hey, this is not right. And the cell would probably undergo apoptosis. But if you have a mutation, then apoptosis probably won't happen. And so everything just goes haywire. Um, and so you just get all of these abnormal cells that eventually just keep dividing, and that leads to cancer. So that's like the overview of proto-oncogenes and oncogenes and like what happens with them and why it can lead to cancer. Does anyone have any questions about that? Yeah, so mutated cells go past the checkpoints. So normally like the checkpoints will stop a mutated cell from continuing through mitosis. But if something is wrong with like either the proto-oncogenes or the tumor suppressor genes, then the cell can move past the checkpoint even though it's defective. Okay, cool.
I have a quick
Liz,
question.
yeah.
So the proto-oncogenes, we have those in our body. That's like normal, right? But when it's mutated is when basically cancer can occur.
Yes.
And the proto-oncogenes, would you say that's like prevents cancer? Because if onco means cancer, wait, or no, sorry, it means tumor. Like Garrett said, that means like, because proto means self, right? So I mean, but it sounds like the proto-oncogenes are like preventing the cancer, but because we have them normally in our body, right? Or are they not preventing?
Yes, they do prevent cancer. I guess I don't know. I don't really, I think Garrett would be better at explaining the like meaning behind the proto part. I don't know. I don't know much about that. Garrett, do you?
Yeah. So the etymology is proto means like before or prior to. So proto-oncogene is something that is a gene that is before it's cancerous. So it's something that can potentially develop into a gene that promotes cancer, like an oncogene, onco, cancer, gene. So a proto-oncogene is just like the variant of a gene that's susceptible to becoming a highly cancerous gene, or a gene that promotes cancer. So it's
Ah,
like a person who's actually, I'm not gonna make that analogy, but yeah, hopefully that makes sense.
yeah, it does. Thank you.
Does anybody else have any questions about that before we move on? Okay, if not, can anybody tell me what a tumor suppressor is? Yes, so it prevents tumors forming by killing defective cells. So like it puts the brakes on cancer, like Casey said. And so basically, this works in conjunction with proto-oncogenes to make sure that normal cells, or sorry, not normal, abnormal cells are going to die and they won't continue moving through the cell cycle. So, what happens if they are mutated? Yeah, exactly. This one is kind of self-explanatory. So basically, if there's a mutation in a tumor suppressor gene, your defective cells are not going to die. And so those defective cells can continue to divide and divide and divide, and they will cause a tumor and cancer will form. What was an important tumor suppressor gene that she had mentioned today? Yes, P53. And I just think that one is interesting because I remember learning about it when I was a student. And it's the one that I think we kind of know quite a bit about. And so that's why she had pointed it out today. And that was all that I had on my end. Like I said, my part was really short compared to Garrett's. Does anybody else want to talk about anything that we went over today or Thursday? Are tumor suppressors regulators? Yeah, you can kind of think of them as regulators in the sense that they control whether defective cells are able to make it through the cell cycle. Oh yes, Garen makes a good point. Tumor suppressors do not regulate oncogenes. Um, oh yeah, so Joseph, there are more than just P53. I just added P53 in there because it's the one that was pointed out today, like specifically. And so I just thought that you guys should be aware of it. But there are like many, many more tumor suppressor genes, I should say, example P53. Oh, so Garrett, Maya asked, can you explain the mitochondrial DNA being a problem with saving a species?
Yes, of course. So what's, mm, well, we know that when you have an embryo, specifically for animals, when you have an embryo that's forming, the fusion of two independent gametes, One is a male sperm gamete, and the other is a female egg gamete. And the sperm gamete is highly reduced, and it doesn't carry like absolutely all the stuff that an organism needs. And that's because it has to move around. There's a lot of selection that went into sexual reproduction. And actually, there's a great video about that I'd love to show you. But yes, so the female gamete, because it doesn't move around, because it's larger, it's known as the megagamete, and it holds the mitochondrial DNA. And humans, endosymbiosis, we need, or we're eukaryotes, and we have mitochondria because that's where cellular respiration takes place. And so obviously we need that, but the sperm doesn't have that. And so we get that exclusively from our mother. My mitochondrial DNA is identical to my mother's. Every lizard's mitochondrial DNA is perfectly identical to their mother's. And mitochondrial DNA is not something that we get from meiosis. Mitochondrial DNA has its own like evolution within our cells and within the cells of all eukaryotes. So if you have, say, or because we know that the mitochondrial DNA only comes from our mother, and we will have identical mitochondrial DNA to our mother because we only get our mitochondria from our mother. If you have a species, for example, the black-footed ferret, that's entirely extinct with no living members, if we put that DNA into a female ferret, that will be its like surrogate mother. If we put that DNA into a surrogate ferret that is not a black-footed ferret, that's maybe a member of a very popular species that is not in any endangerment of endangered species. There is a better way to say that. But yeah, so if you put highly endangered DNA into a surrogate mother that's not endangered, then the individual is, I just activated my Siri by saying that, then the individual is going to have the mitochondrial DNA of the surrogate mother. And so the mitochondrial DNA that existed in the very rare, now extinct DNA of the black-footed ferret is absolutely gone, never to be recovered. Unless you can somehow impregnate a male, then any like resulting species from the cloning is going to get the mitochondrial DNA of the surrogate mother, which is going to be a very present species that have a lot of individuals, because only the mitochondrial DNA was injected into only the nuclear DNA was injected into the surrogate mother. How was it lost? Because even though we had our like the last living individual of the black-footed ferrets was a female named Willa, and Willa obviously died without reproducing, but we had her DNA preserved. We had a bunch of her cells preserved. And yes, mitochondrial DNA does exist within all those cells, but only the nuclear DNA was taken from Willa's cells and injected in vitro into the surrogate mother, not the mitochondrial DNA. And even if the mitochondrial DNA was injected in, actually, I can't even comment on what would happen. I have no idea whether or not the mitochondria would like just instantly die, or whether or not they'd have to compete with each other, or I don't know. But yes, Maya said that makes sense. It was lost because only nuclear DNA was injected, and mitochondrial DNA does help with genetic diversity. Absolutely. Mitochondria, like I said, they are their own independent organisms pretty much within every single one of our cells. And you cannot overstate how important they are, because they provide all of the energy that our cells utilize for all their chemical processes. We took 213 and we learned about cellular respiration. And so some mitochondria, like the mitochondria within my cells, are different than the mitochondria within an ant's cells. And ant mitochondria are going to be able to utilize or undergo cellular respiration at a different level of efficiency than mine are. because I'm an endotherm and they're an ectotherm, or maybe reverse that. I don't remember. But yeah, different mitochondria, different organisms, different capability. So yeah, genetic diversity is impacted by mitochondrial DNA.
I have a question on that. So if the nuclear DNA was injected into the surrogate mother from the extinct species, nuclear DNA was injected into the surrogate mother, I'm guessing for the purpose of like kind of trying to prolong the species that's already extinct.
Mm-hmm.
Well, then how or why does I understand why the mitochondrial DNA wouldn't be present in the surrogate mother or the offspring, because that was not the DNA injected, but what about the nuclear DNA? Is that at least present in the offspring?
Yes, absolutely. That's the whole point. And I suspect it would be like the same amount of present. Willa's DNA within Elizabeth Ann would be at just the same like level of presence as like your father's DNA would be in your DNA, if that makes sense. So Willa, even though she wasn't able to donate her mitochondrial DNA, donated just as much nuclear DNA as any parent of any animal. But an interesting question is, do we think that the genes that are subject to a maternal effect would be exhibit the maternal effect of Willa's DNA or the surrogate mother's DNA? That's a good question. Which I don't know the answer to, so don't ask. But thank you for that question. That was a good question. All very good questions. Does anyone have any more questions? I make myself laugh. I also dropped my internet connection like 30 seconds ago. Can you still hear me? Okay, no more questions. Thank you.
No, we can still hear you.
Another example of the COC. I'm unfamiliar with this acronym.
What is coefficient of?
Oh, okay.
Oh, okay. I don't have a question prepared, but if you have a quiz, I know that there's some in the textbook. If you want to pull one of those up, we could do it. There should be, Garrett, we did one last review, but we also did one the previous review. So the last two reviews both have questions on it.
Mm-hmm.
Oh yeah, Caroline, if you have, is it from quiz 8 or from a previous quiz? Quiz 8. Okay, so we can't talk about quiz 8 until quiz 8 closes. Or quiz 8 did close already, didn't it? Right?
Mm-hmm.
I can't remember what number quiz we're on anymore. Okay, yeah. So then, yeah, Caroline, which one do you want to go over?
It was the one about recombinant frequencies.
Okay, let me see.
I think it would be Maya. That would make sense.
Is it given the following data, find the recombination frequencies, and it's like WXS and then a bunch of numbers and stuff?
Yes.
Okay, so that is literally like the exact same question that Garrett and I went over during our review.
Okay, I'll go back and look at it then.
We can go over it again, but like we talked about it
Yeah.
quite extensively during not last week's review, but the week before. Let me pull it up. Oh yeah, it was identical. So let me share my screen. Yeah, so it was same number, same question. It was this question, right?
Yeah. For that review, I did have to leave early, so I might have just missed it.
Okay, yeah. So basically I'll give you like quick rundown. So basically what you have to do is you have to find out which one is parental and which one is your double crossover. And so parental is always going to be your biggest group and double crossovers are always going to be your smallest. And then you compare your double crossovers to your parents, and whichever gene doesn't match is going to be your middle gene. So we found that it was S. And then for recombination frequencies, you have to find it between your middle gene and one of the other two genes, and the middle gene and the other gene. So we did SX and SW because S was the middle, and then we have to compare it to X and compare it to W. And all you do for SX is find which of your progeny have different SX, which one have different S's and X's from your parents, and then divide it by the total, and that'll give you that recombination frequency. And you do the same thing for SW. And then Carlos kind of talks about coefficient of incidence also. So basically what you do is you take the number of your actual recombinants and then divide it by what you expected. So what you expect is the individual, sorry, someone was calling me. So you take your two recombination frequencies that you found and multiply them together, and then multiply it by your total. And when you divide those two, you get your coefficient of interference. And then interference is just 1 minus the coefficient. And that is what we found here. And if you go to the very bottom of this document, there is a step-by-step guide that walks you how to solve a double crossover question.
Thank you so much. It
Mhm.
cleared it up. I was just confused on how you chose the certain ones to put into the equation, but now I get it.
Okay,
There's
cool.
also a video. Oh, sorry to interrupt. There's also a video if you want to explore it some more.
Yes, Garrett made a very, very helpful video. I 120% would recommend that you watch it before the exam because it walks you through double crossovers very, very, very nicely. Carla, do you still want to go over how to find the middle gene and like why it's S? Okay, so basically what you have to do, I'm going to do this because we know that this is the top two are our parentals, and the bottom two are our recombinants. And so basically what happens is we have to compare our recombinants to our parentals. And so we can compare W plus X plus S to our parents, and we can see that it's pretty close to W plus X plus S plus, but not quite. The S is different. And then also when you do the same thing with WXS plus, and you compare with WXS because they're the closest, but they're not quite the same because the S is still different. And that difference tells us that the middle gene is S. Yes, so S is the only one that differs in both of them, and that's why it's our middle gene. No problem. Is there anything else anyone would like to go over?
If you don't mind, since you have this open, can you just walk us through super quickly how you get the numerator for the recombination frequencies? I know that the double crossover is always included, but 74, 91, and then 83 and 78.
Oh, sorry. So, like why 91 and 74 are in this calculation, why 78 and 83 are in this one?
Yes.
Okay, so we are going to cut and paste again. So basically what we do is we always go back and compare to our parental. And so what we want to do is we're looking for the distance between, or sorry, whether there was recombination between S and X. And so if we look at our 91 and 74, when you compare 91 to both of your parents, you can see that it's most closely, it looks most similar to 356, but there's something that's kind of funky about it. And the funky thing is that instead of X plus S plus, it's XS plus. And so that tells us that there was crossing over between S and X because those two were different from our parent. And then when you look at 74 and both of our parents, you can see that it looks most like our 374 because we have W and W and S and S. But if you look at the X's, you can see that you have X and then X plus. So similar, but not the same because of that difference between the X's. And so because of that, that means that you have crossing over between X and S. But if you do the same thing for our other group, you'll see that when you look, so right now we're comparing just S and X. So if you compare 78 to 374, because they are the most similar, but not quite. If you look at our S and our X's, they are identical. They're both SX. And the reason why you can't compare 78 to 356 is because it differs in two places. You have W plus and W plus, which are the same, and it's fine. But then you have X plus X and S plus S. And so that's why you don't have crossing over in 78. And for the same reason in 83 when it comes to SX. And then you can apply the same thing for SW. Does that make more sense now?
Yes. Thank you.
Does anybody else have any questions they'd like to go over? Yes, there's going to be another review on Monday at 7:30 p.m. Yeah, we can review textbook questions, but like you guys have to ask us. Like we do say that every time. If you have questions, just ask us about them, but we don't know which ones you have questions about if you don't ask. Yeah, Casey, it can be from anything. And like if anytime when you guys are like doing the homework questions on your own, if you want the solutions, you can just email Garrett and I, and we'll send you the solutions for the questions. Crystal, we just went over that. basically you have to compare all of your genotypes to your parentals, and wherever they differ and they're not the same as your parentals is going to be part of your recombination frequency. Yes, basically watch Garrett's video. Like, it is so helpful. I will literally tell you where to find it right now. If you go to LA resources on the side, recordings and documents for exam 3, and then you go, where is it? Oh, 3-point cross practice. It is the second video down, and it is literally titled 3-point cross practice. And yes, it is an hour, but you can probably watch it on 2 times speed, and then it would be 30 minutes. And it goes over it really well. And we talk about it also in this weekly review, the first video link that says weekly review week 10. And we go over 3-point cross for like 30 minutes. And then there's also this really great summary at the end of this document, which can also be found attached right here. So there is a lot of 3-point cross practice and information.
There's also an example in trans on the week 10 review, question mark, last week's review.
Yes, last week's review, this one.
We're in week 12 already. Oh my gosh.
No problem, guys. Is there anything else that people would like to go over besides 3-point crosses? Slide 23 from chapter 11, sure. Let me see. All right, so slide 23. Is this the slide that you were talking about, Casey? Okay. So what did you want to talk about from this slide? Or just like the whole slide in general?
So, let me see. I wrote my notes on this slide, so I'm pulling them up. I guess I just want to make sure I have the key takeaway correctly, which basically what I got from this slide when I listened to the lecture was that during life cycle and development of the chicken, when looking at the different globin genes, the genes are expressed by those little tiny red arrows at different times during the development. And and also she said something like, when you have transcription, you don't transcribe the entire genome, only the gene of interest. And she had said, I guess specifically looking at chromatin changes, you see changes here when you have embryo at 5 days old, and also changes when the embryo is at 14 days. And the quote unquote change means, like, we're talking about all the tightly packed chromatin is unpacked and available for transcription.
Yeah, I think that you got most of it, like what you should take away from the slide. So basically, yeah, what happens is you don't transcribe all of the genes all at once. Like you said, you only transcribe the ones that are of interest at the moment. And then what happens is after X amount of days, the embryo all of the chromatin in the embryo unwinds. And so all of it is getting transcribed all at once. And so at that point, all of the DNA is sensitive to DNase 1 because all of the chromatin is unwound and loose and is exposed. So DNase can act on it, but before and after those points, or sorry, before the points where all of the DNA, all of the chromatin is unwound and being transcribed, only the parts that are being targeted and transcribed at the moment would be sensitive to DNase.
Okay. All right. Yeah, it says, it says at the top is chromatin structured altered in transcription. Yeah, and I guess that's like you said, the tightly packed chromatin's unpacked available for transcription, but not all at the same time, just for whichever one is being whatever the gene is of interest.
Yes,
Cool.
exactly.
All right, thanks.
All right, we wanted to have this be kind of a shorter review. So if there are any burning last minute questions, please ask them so we can maybe move on a little bit sooner.
One more question from me. Do you think that we talked about barcoding very briefly, and trying to find my notes on it. I just had my slide pulled up. I know you don't know what's going to be on the exam, but I just wanted to make sure I understood what barcoding was or is. Sorry, I just had it pulled up and now I can't find it. Okay. So barcoding is where we collect samples, extract the DNA, sequence a standard piece, then compare with what's available already from that species to understand the species found and if it's like already in existence or if it is a new species found.
Oh, wait, sorry, can you say that again?
Yeah, sure. So I understand that barcoding is basically where to screen biodiversity to get an idea if you're finding new species. So you collect samples from the environment of interest, extract DNA, and then barcode, meaning sequence standard piece, like a specific gene, and then compare with what's available already from organisms that we know. So you can have an idea of how different the organism is, that if what you found is like compare it to what already exists.
Yes, that honestly, that was all I understood from the barcoding also. Okay. So I don't know if Garrett could go more into depth.
I don't even know what barcoding is. Sorry.
But yeah, no, that's all I was able to like extrapolate and take away from barcoding when we talked about it also. So I think that should be like as much as you should know about it, hopefully, because I can't imagine that Dr. Chiari would go like much more into depth about it than that.
Okay, cool. And then also a core histone, that is 8 histone proteins, right?
Yes. Okay. So the core is 8, but then there's a 9th one that latches your chromatin on the outside. So one nucleosome has 9 total histones, 8 in the middle, and then 1 on the outside that holds the chromatin on.
Okay, you said a nucleosome is 8 histone proteins. That's like the core histone. And then that ninth one, does that ninth one make it a chromatosome though? Because it's that histone H1 that locks the DNA in place, like you said. Does that make it entirely, does that change it from a nucleosome to a chromatosome?
That's a good question. I don't think
I'm looking
so,
at
Garrett,
slide-
does it?
It's on slide 17 of chapter 11.
I always thought that a nucleosome was. Okay, yeah, then I guess I was wrong. So you're right. A nucleosome is just the 8 histones, and then a chromosome is 9 histones. I'm sorry.
Okay. Got it.
Yeah, we learned in 213 that a nucleosome is the 8 histone proteins that have DNA wrapped around them, and that's the like basic unit of DNA coiling.
Mhm. All right. If anyone else has any questions, speak now or forever hold your peace.
Thanks for hanging out with us and sticking around for so long. It's only been an hour though. Okie dokie. Well, hope you guys have a great rest of your day. The birds are singing and the wind is blowing. It's fantastic.
All right. We will see you guys then. You guys are free to go.
Yes. Bye, everyone.